All right, this is part 3 of your cell respiration notes. So I'm going to start this set of notes with a brief overview of what we did last time. Basically, here's our cell, here's the mitochondria, and we started with glucose that you get from consuming food that gets broken down into two G3P molecules, right, that are each made of 3 carbons. Those G3P molecules are further modified into pyruvate, which is also a 3-carbon molecule. Those 2 pyruvates that you make from glucose then move into the mitochondria, where they run through a series of reactions that changes the amount of carbons present. You basically rip off all the carbons to make carbon dioxide that you then exhale because that diffuses out of the mitochondria, out of the cell, into the bloodstream, into the alveoli in your lungs, and then out of your breath, right? Every time you do that, you're breathing out carbons in the form of carbon dioxide that came from glucose. So if you had like cereal for breakfast or something, and maybe like a couple hours later you're exhaling, the carbons that you're exhaling are the same carbons you digested as glucose a couple hours earlier. I know, isn't that cool? Anyway, so we made all of those carbons, and because we break bonds to release those carbons, every time we broke a bond, we released an electron, and that electron was captured by an electron carrier. And we made 10 of those carriers in the Krebs cycle. We made 2 of them in glycolysis, and that matches the 12 hydrogens that we had to start with in our glucose molecule. Right. So what happens now? Where are we going now? And that means that we have to continue this story by looking at the matrix and this folded membrane of the mitochondria. So we talked about these 3 sets of proton pumps that occur as a triplet in this folded membrane, this inner membrane. Remember, you have the cell membrane, then you have the outer mitochondrial membrane, you have the inner membrane space, and then you have this folded inner membrane that has this triplet of proton pumps. And these were the names of them, right? When we went over the names last time, and they pump protons. All right, so that's where our story takes us. So let's start with where we left off. We left off with the mass production of electron carriers. We had 8 NADHs and 2 FADH2s per glucose. So we're doing this thousands of times. There's not just one Krebs cycle in a mitochondria. There's many Krebs cycles. So there's a constant supply of NADH being produced in the matrix, diffusing away from it, diffusing out towards the periphery of the mitochondria, which means it's diffusing out towards this folded inner membrane, right? Well, each of these proton pumps is increasingly electronegative, right? Cytochrome C is more electronegative than cytochrome BC complex, which in turn is more electronegative than the NADH dehydrogenase, which in turn is more electronegative than NADH. So NADH is going to diffuse out towards the electron transport chain, and it's going to drop off the electron. I'm going to do a little circle here for the electron, and it's going to leave as NAD+. It's going to diffuse as NAD+ back towards the middle of the matrix, where it can then proceed to pick up more electrons that are going on in the Krebs cycle. Right, so that's kind of the movement of these electron carriers. So once it's been reduced, they will diffuse towards the electron transport chain, and then once they've been oxidized again, they diffuse back into the matrix. So this is just random diffusion. Once the electron has been dropped off, what is it going to do to this protein pump? What is it going to do to the shape of this protein pump when you just drop a negative charge in it? Well, it's going to make the shape change. It's going to cause a conformational shape change in this protein. And that is going to cause, remember, these are proton pumps. That's going to cause it to pump, oops, I drew my arrow a little bit too far. It's going to cause a proton to get pumped into the inner membrane space. And you're like, well, where did that proton come from? Well, the matrix has a pH, right? The matrix has actually a pH of about 7, which means its concentration of protons is times 10 to the negative 7 molar. So you have free-floating protons in here as kind of the default in the matrix. Those are the protons that are getting pumped into this inner membrane space, right? Well, because the proton pumps in the electron transport chain are increasingly electronegative, this electron is going to get passed. This is the hot potato that we talked about back in the first video. They're going to get passed on to the next electronegative proton pump in this chain. And that's going to reset NAD dehydrogenase so that it's ready to receive a new electron, right? So the electron gets passed. That's going to cause a shape change, just like it did for NAD hydrogenase. It causes a shape change in cytochrome BC complex, right? And it pumps, you guessed it, it pumps a proton through. Well, cytochrome C is even more electronegative, so the electron gets passed one more time. That causes a shape change in cytochrome C. A proton gets pumped, and cytochrome BC gets reset. So what we're doing is we're pumping electrons into this inner membrane space using an electron rather than ATP as the energy source. And that actually, I'm going to go ahead and just add a bunch of protons in this inner membrane space. The entire inner membrane space is full of these electrons. So there's electron chains all over the place. It's not just the one set of 3, they're everywhere. So this entire inner membrane space is getting pumped full of protons, and the pH of the inner membrane space is about 5. So there's about 100 times more protons in this space than there is out here in the matrix, right? Before we turn to the electrons after the ETC, let's look at what happens to these protons. Well, these protons, because they've been pumped into this inner membrane space, really are going to want to diffuse back into the matrix from a high concentration in the inner membrane space to a lower concentration in the matrix. But they can't go right through this membrane. They can't go through that folded inner membrane because they are positively charged, and this membrane is a phospholipid bilayer that's going to prevent charged molecules, charged ions from doing so. So what they have to do is they have to facilitatively diffuse. They have to diffuse with the help of a protein channel. And this is where ATP synthase comes into play. ATP synthase. ATP synthase is pretty cool. Not only is it a channel for these protons, I think I might have said proteins when should have been saying protons. These protons, not proteins, diffuse. But ATP synthase is a protein. It is not only a channel for these protons, it is also an enzyme. It's a dual purpose protein. It's also an enzyme. So as these protons diffuse through ATP synthase, and the shape of ATP synthase does matter. The kind of the bodily portion is in the matrix, and the neck is kind of sticking out into the inner membrane space. So when you draw this, make sure you're drawing it facing the right way. It is going to phosphorylate ADP. You're phosphorylating ADP, and you are using, you guys remember, when this positive charge passes through ATP synthase, a full-on positive charge is moving right through the middle of a protein. That's going to cause it to shape change. It's going to cause the active site and the whole enzyme to change shape. It's like, whoop, there goes the charge, and I'm going to go ahead and change shape. That shape change is going to provide the energy to phosphorylate, or the movement of that charge is going to provide the energy to phosphorylate ADP into ATP. And for every glucose that gets broken down in this entire process, you pass through enough protons through ATP synthase after all of this has been done to make 28 to 32 ATP. And this is the big ATP payoff. This is where most of your ATP is coming from, right? So again, these protons are diffusing. This is facilitated diffusion through ATP synthase, which causes a shape change in ATP synthase that powers the phosphorylation of ADP into ATP. And one glucose has enough of this to make 28 to 32 ATP when all is said and done. So there's your payoff. All right, let's return really quick and finish the story back up here. So we left off saying how these electrons have been kind of passed around through the electron transport chain. But we didn't say what happened to the electron once it was at cytochrome C, because 1 and 2 here are reset, but 3 is not reset. And as long as this electron is parked here, this series of proton pumps is going to be essentially inactive. So we have to get rid of this electron somehow. And what's more electronegative than any of these pumps? What's the most electronegative thing in biology, basically? And that's oxygen. That's oxygen. And this is the whole reason you need oxygen. You need oxygen. You inhale it so that it can serve as the final carrier for this electron. And remember, oxygen can handle 2. So it's going to say, give me that electron, I can handle it, and it can do this twice. And because you have free-floating protons in the matrix, well, they're going to say, hey, buddy, I'm attracted to you. And they're going to join up with this electron. And what have you just made? What's a proton and an electron? Right? That's a hydrogen. You've made water. You've made water. Right? So this kind of water is actually made by your cells. You don't have to consume the water at all. You're actually making it. Actually, the oxygen that you inhale gets reduced into water. That's the whole point of the oxygen you breathe. It's to be an electron carrier. And if you if you hold your breath and you deprive your cells of oxygen, first of all, the feeling of suffocation that you get is actually from a buildup of CO2. But what's also happening in your mitochondria is that your electron transport chain has shut down. There's not enough oxygen to carry all those electrons. So you get a backup of NADH basically, and you can't produce any more ATP because there's no more protons to diffuse back into the matrix. So this is why oxygen is so important in organisms that do aerobic respiration. And this is the only part of the whole process that's aerobic. All right, this is the whole point of aerobic respiration here. This is the only part where oxygen comes into play. But this water that you make is called metabolic water. This is called metabolic water. Metabolic water. This is the same water that a lot of desert animals rely on when they are in the desert. And they're adapted to basically rely on just this kind of water because natural water to drink is hard to come by. All right, let's zoom out. I'm going to draw your attention to some vocab. On the back of the big sheet, we defined substrate level phosphorylation last time, but directly underneath that, there's something called oxidative phosphorylation that we're going to talk about first. We'll do photophosphorylation later. So oxidative phosphorylation is the combination of two things. Let me zoom in here. It's the combination of two things. It's the combination of the electron transport chain, and that is going to, so electron transport and pumping protons, and that creates a gradient, creates a proton gradient. It's a combination of that and what's something called chemiosmosis. Chemiosmosis. And chemiosmosis is just the diffusion of ions. In this case, it's a hydrogen ion that powers ATP synthase. So oxidative phosphorylation is the combination of those two things. So over here, we can write the full definition. We're going to make ATP using ATP synthase and the hydrogen gradient created by the electron transport chain using the electrons from glucose. Right. And actually up here there's a space for you to define chemiosmosis. So remember, we can just define all these terms. Diffusion was any molecule moving down its concentration gradient, right? So moving from high to low concentration. I'll just do that. Use brackets for concentration. Remember, osmosis was specifically the diffusion of water. And chemiosmosis is the diffusion movement of ions, specifically ions, from high to low concentration, right? And for us, in cell respiration, the ion was hydrogen. Right. So those two things, the electron transport chain and chemiosmosis, is oxidative phosphorylation. So back on the other side of the big sheet, here's the electron transport chain up here. We're making a proton gradient. We're pumping protons into the intermembrane space that are then diffusing down through ATP synthase to turn a channel pump, remember it's a dual function, which is going to phosphorylate ADP into ATP. Right, so here's the electron transport chain component, and here is the chemiosmosis component, the diffusion of the ions. This entire thing is oxidative phosphorylation. And remember, the protons are here because they were pumped using the energy that was dropped off to them by these electron carriers, using an electron. Well, the electrons came from breaking the bonds between carbons and hydrogens in the Krebs cycle. Well, the carbons and hydrogens came from pyruvate out in the cytoplasm, which came from the glucose that you consumed. Right? So big picture, the glucose that you eat gets broken down, all of the carbons get ripped off and exhaled using in the form of carbon dioxide, right? And the electrons that are released when you break those bonds get loaded into electron carriers. They drop off the electrons in the electron transport chain. Those electrons are passed like a hot potato down to the oxygen that's only there because you've breathed it in to form water. Protons are pumped into the intermembrane space, which then diffuse back through a channel enzyme to make ATP. This is the ATP that you get from glucose. Remember, you can't really store ATP, so ATP is really unstable because it's basically a spring-loaded molecule that's ready to burst apart and give away that third phosphate, right? So you kind of have to make this on demand. You can't really store this very easily. So these are made on demand right then and there. When your cells need energy, right? So that's the big picture. We will discuss a little bit more on this big sheet, including the section down here later. And we'll also talk about some feedback loops in class. So I hope this helped. You don't have to memorize any of this. You don't have to memorize any of the names. You don't have to memorize where the carbons are lost or where the NADs or FADHs come into play. But you should have an appreciation for the overall picture, right? Overall, what's happening to the carbons, where are the what's happening to the electrons? You're passing the electrons to electron carriers. The electron carriers drop off the electrons at the ETC. The whole point of that's to pump protons. The protons diffuse back into the matrix through ATP synthase, and you mass produce ATP. So the big picture is what's important, not necessarily the nitty-gritty details. And that's what I want you to focus on. I don't want you to get overwhelmed with all the specifics. If you get the big picture, you should be just fine. So we'll continue with this in class, and I will see you later.